Biochemistry. Cellular Part 1. Cell cycle phases. The checkpoints control transitions between phases of cell cycle. This process is regulated by cyclins, cyclin-dependent kinases, and tumor suppressors. M phase is the shortest phase of cell cycle, which includes mitosis and cytokinesis. Mitosis include prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis is the cytoplasm splits in two. G1 is of variable duration. Regulation of cell cycle. Cyclin-dependent kinases. Cyclin-dependent kinases are constitutively expressed but inactive when not bound to cyclin. Cyclin-CDK complexes. Cyclins are phase-specific regulatory proteins that activate cyclin-dependent kinases when stimulated by growth factors. The cyclin-CDK complex can then phosphorylate other proteins, for example, retinoblastoma protein, to coordinate cell cycle progression. This complex must be activated or inactivated at appropriate times for cell cycle to progress. Tumor suppressors. Tumor protein p53 leads to p21 induction, cyclin-dependent kinases inhibition, Rb hypophosphorylation, activation, and finally G1 to S phase progression inhibition. Mutations in tumor suppressor genes can result in unrestrained cell division, for example, Li-Fraumeni syndrome. Growth factors bind tyrosine kinase receptors to transition the cell from G1 to S phase. Growth factors include insulin, platelet-directed growth factor erythropoietin, and epidermal growth factor. Cell types. Permanent type. Remain in G0, regenerate from stem cells. Permanent cells include neurons, skeletal and cardiac muscle, and red blood cells. Stable quiescent type. Enter G1 from G0 when stimulated. Stable cells include hepatocytes, lymphocytes, proximal convoluted tubule, and periosteal cells. Labile type. Never go to G0, divide rapidly with a short G1. Labile cells are most affected by chemotherapy, which include bone marrow, gut epithelium, skin, hair follicles, and germ cells. Rough endoplasmic reticulum. Site of synthesis of secretory exported proteins and of N-link oligosaccharide addition to lysosomal and other proteins. Nicel bodies. Rough endoplasmic reticulum in neurons. Synthesize peptide neurotransmitters for secretion. Free ribosomes are unattached to any membrane. Site of synthesis of cytosolic, peroxisomal, and mitochondrial proteins. N-link glycosylation occurs in the endoplasmic reticulum. Mucus-secreting goblet cells of small intestine and antibody-secreting plasma cells are rich in rough endoplasmic reticulum. Proteins within organelles, for example endoplasmic reticulum, Golgi bodies, lysosomes, are formed in rough endoplasmic reticulum. Smooth endoplasmic reticulum. Site of steroid synthesis and detoxification of drugs and poisons. Lax surface ribosomes. Location of glucose 6-phosphatase. Last step in both glycogenolysis and gluconeogenesis. Hepatocytes and steroid hormone-producing cells of the adrenal cortex and gonads are rich in smooth endoplasmic reticulum. Cell trafficking. Golgi is the distribution center for proteins and lipids from endoplasmic reticulum to vesicles and plasma membrane. Post-translational events in Golgi include modifying N-oligosaccharides on asparaginine, adding oligosaccharides on serine and threonine, and adding mannose-6-phosphate to proteins for targeting to lysosomes, usually for degradation. Endosomes are sorting centers for material from outside the cell or from the Golgi, sending it to lysosomes for destruction, or back to the membrane or Golgi for further use. Eye cell disease, also called inclusion cell disease or mucolipidosis type 2. Inherited lysosomal storage disorder, autosomal recessive. Defect in N-acetylglucosaminal 1 phosphotransferase leads to failure of the Golgi to phosphorylate mannose residues on glycoproteins, with decreased mannose 6-phosphate. enzymes secreted extracellularly rather than delivered to lysosomes. Lysosomes deficient in digestive enzymes, which leads to buildup of cellular debris in lysosomes, inclusion bodies. Results in coarse facial features, gingival hyperplasia, corneal clouding, restricted joint movements, claw-hand deformities, kyphoscoliosis, and increased plasma levels of lysosomal enzymes. Symptoms similar to but more severe than Hurler syndrome, often fatal in childhood. Signal recognition particle, abundant cytosolic ribonucleoprotein that traffics polypeptide ribosome complex from the cytosol to the rough endoplasmic reticulum. Absent or dysfunctional signal recognition particle leads to accumulation of protein in cytosol. Vesicular trafficking proteins, COP1, Golgi to Golgi, retrograde, cis-Golgi to endoplasmic reticulum. COP2, endoplasmic reticulum to cis-Golgi, anterograde, 2 steps forward, 1 step back. Clathrin, trans-Golgi to lysosomes, plasma membrane to endosomes, receptor-mediated endocytosis. For example, LDL receptor activity. Peroxisome, membrane-enclosed organelle involved in beta-oxidation of very long-chain fatty acids, strictly peroxisomal process. Alpha-oxidation of branch-chain fatty acids, strictly peroxisomal process 2. Catabolism of amino acids and ethanol. Synthesis of bile acids and plasmalogens. Important membrane phospholipid, especially in white matter of brain. Zellweger syndrome. Autosomal recessive disorder of peroxisome biogenesis due to mutated PEX genes, which leads to accumulation of pipolic acid in peroxisomes. Clinical features include hypotonia, seizures, jaundice, craniofacial dysmorphia, hepatomegaly, and early death. Refsum disease. Autosomal recessive disorder of alpha oxidation which leads to buildup of phytanic acid due to inability to degrade it. Clinical features include vision loss, retinitis pigmentosa, anosmia, hearing loss, ataxia, peripheral neuropathy, ichthyosis, and cardiac conduction defects. Treatment is diet and plasmapheresis. Adrenoleukodystrophy. X-linked recessive disorder of beta-oxidation due to mutation in ABCD1 gene, which leads to very long-chain fatty acids buildup in adrenal glands, white matter of brain, and testes. It is a progressive disease that can lead to adrenal gland crisis, progressive loss of neurologic function, and death. Proteasome. Proteasome is a barrel-shaped protein complex that degrades polyubiquitin-tagged proteins. It plays a role in many cellular processes, including immune response, major histocompatibility complex 1 mediated. Defects in ubiquitin proteasome system are also implicated in diverse human diseases, including neurodegenerative diseases. Cytoskeletal elements. Cytoskeletal elements are a network of protein fibers within the cytoplasm that support cell structure, cell and organelle movement, and cell division. Microfilaments, for muscle contraction, cytokinesis, and phagocytosis. Examples of microfilaments are actin and microvilli. Intermediate filaments. Intermediate filaments maintain cell structure. Examples of intermediate filaments are vimentin, desmin, cytokeratin, lamins, gliofibrillary acidic protein, and neurofilaments. Microtubules, for movement and cell division. Examples of microtubules are cilia, flagella, mitotic spindle, axonal trafficking, and centrioles. Microtubule. Cylindrical outer structure composed of a helical array of polymerized heterodimers of alpha and beta tubulin. Each dimer has 2 GTP bound. Microtubules are incorporated into flagella, cilia, and mitotic spindles. Microtubules are also involved in slow axoplasmic transport in neurons. Molecular motor proteins. Molecular motor proteins transport cellular cargo toward opposite ends of microtubule, retrograde to microtubule, from positive to negative. Dynein. Anterograde to microtubule, from negative to positive. Kinesin. Clostridium tetani toxin, poliovirus, rabies virus, and herpes simplex virus use dynein for retrograde transport to the neuronal cell body. Herpes simplex virus reactivation occurs via anterograde transport from cell body, kinesin-mediated. Slow anterograde transport is the rate-limiting step of peripheral nerve regeneration after injury. Drugs that act on microtubules. Microtubules get constructed very terribly. Antihelminthic mebendazole, antifungal grisolfovin, antigout colchicine, anti-cancer vinca alkaloids, and anti-cancer taxanes. Negative end near nucleus, positive end points to periphery. Ready, attack. Cilia structure. Motile cilia consist of 9 doublets plus 2 singlets arrangement of microtubules. This microtubule-based cytoskeleton, also called the axoneme. Basal body is the base of cilium below cell membrane. Basal body consists of 9 microtubule triplets with no central microtubules. Non-motile primary cilia work as chemical signal sensors and have a role in signal transduction and cell growth control. Dysgenesis may lead to polycystic kidney disease, mitral valve prolapse, or retinal degeneration. Axonamel-dynein. Axonamel-dynein is an ATPase that links peripheral 9 doublets and causes bending of cilium by differential sliding of doublets. Gap junctions enable coordinated ciliary movement. Primary ciliary dyskinesia. Primary ciliary dyskinesia is an autosomal recessive disease. Dynein arm defect leads to immotile cilia, dysfunctional ciliated epithelia. The most common type is Carter-Jenner syndrome. Primary ciliary dyskinesia with situs inversus. Pulmonary ciliary dyskinesia presents with developmental abnormalities due to impaired migration and orientation. Situs inversus. Hearing loss due to dysfunctional eustachian tube cilia. Recurrent infections. Sinusitis. Ear infections, bronchiectasis due to impaired ciliary clearance of debris and pathogens, infertility, increased risk of ectopic pregnancy due to dysfunctional fallopian tube cilia, and immotile spermatozoa. Lab findings include decreased nasal nitric oxide, used as screening test. Sodium-potassium pump. Sodium-potassium ATPase is located in the plasma membrane, with ATP site on cytosolic side. For each ATP consumed, 2 potassium go into the cell, pump dephosphorylated, and 3 sodium go out of the cell, pump phosphorylated. 2 strikes, K, you're still in. 3 strikes, nah, you're out. Digoxin directly inhibits sodium-potassium ATPase, which leads to indirect inhibition of sodium-calcium exchange, increased intracellular calcium concentration, and increased cardiac contractility.